Good afternoon. So my name is Jeremiah Fasano. I'm from the Center for Food Safety and Applied Nutrition at FDA, and I'm going to talk a little bit about our biotechnology consultation process. I think most of you are probably already very familiar with this, so I thought what I'd try to do today is, rather than focus on the minutiae of the process and sort of mental checklists you may have or that we may have, I think it's human nature to have those, to talk about sort of the fundamental principles underlying this process, because that's really the touchstone for the consultation process. It's not in code and regulation. As was mentioned earlier, it's sort of an outgrowth of the coordinated framework, but it really is a principles-based process, and so that's what I'm going to talk about today. So the U.S. food supply is extremely large and complicated, as you all know, and it's FDA's responsibility to make sure that foods and feeds in the U.S. food supply are safe. And since the 1990s, foods and feeds from genetically engineered food crops have been part of that food supply, and so they are part of our responsibility to make sure that they're safe. We have a consultation process. It is voluntary. And the purpose of this process is to allow FDA to engage with developers of genetically engineered food crops to make sure that all their responsibilities under the Food, Drug, and Cosmetic Act are fulfilled. Once we complete the consultation, we post it on our website. And I think the consensus has been, whatever anybody's feelings in any particular case, that in general this process has been very helpful both for FDA and for biotech industry. The question that frequently comes up, and this is why I was sort of alluding to checklists earlier, is what kind of information is needed to complete the process? Well, the bottom line, as you've already heard, is that we consider this a case-by-case process. And so the particular information that's going to be needed is going to be highly dependent on the traits that you're introducing, the crop that you're introducing them to, and the specifics of the transformation events involved. There obviously are a number of types of data that you frequently see. I've put some of them up here, but fundamentally it's really about what is relevant for that particular crop and event and trait. We do encourage people to come in early and often. Part of the reason for this is so that we can provide feedback on what we see as most appropriate for any particular crop. But what we're really interested in, sort of the touchstone here, is information that's relevant to safety. And that's framed in terms of two key questions that I'm going to talk about in a little bit more detail. So this is a very flexible process. It really is case by case. It's dependent on specific properties of the crop and the transformation event. And we feel that that flexibility has been extremely useful, again, both for FDA and for protecting public health. You've already heard about the coordinated framework, so I'm going to skip over this slide. The fundamental point here is just that it's FDA's responsibility to make sure that foods and feeds from genetically engineered food crops are safe, just as all other foods and feeds are required to be safe. So there's actually, although you wouldn't know it to look at this slide, a fairly simple way to think about what FDA is interested in. Yes, just ignore the flowchart. It's the 1992 policy. This is a statement of policy that we published in 1992 explaining how FDA's existing authorities would be used under the coordinated framework for the regulation of genetically engineered food crops. And the key thing that we said in this policy statement was that we were really interested in the objective characteristics and components of any genetically engineered food from some genetically engineered plant, and not necessarily in the particular process that was used to develop that crop. And we also explained in the 1992 policy how we would use our existing authorities and how they would apply in considering the products of genetic engineering. That really comes down to two key questions. The first one is, what have you added to the food? Now, in this particular image, we're talking about a protein, but of course it doesn't have to be a protein. It could be one protein, could be multiple proteins, either because you're building some kind of metabolic pathway, you're introducing a marker, you have multiple traits. It could be a transcription factor, as Wayne had alluded to earlier, or genetic material regulating the expression of existing proteins in the plant. And then the other question we have is, what else happened when you were doing that? Right? This is sort of the unintended effects piece. What happened to the plant when you're introducing your intended effect? And so those two key questions are really what are in our minds as we're considering a new transgenic crop. This is the one really regulatory slide, just to talk about our authority. And this is important just because it'll help you understand why we focus on these two key questions. So FDA's authority is to make sure that foods and food ingredients are safe. Our food ingredient authority comes from the food additive provisions of the Act, Section 409. We also have authority over whole foods and finished foods, including unprocessed foods like produce, under the food adulteration provisions of the Act, Section 402. So together, these are some key regulatory tools we have for making sure that foods and food ingredients are safe. And so these sort of frame and give force to these two questions we ask about what did you add and what else happened while you were doing that. In terms of that first question, in the 1992 policy, we explain that any deliberate change that is being made to a genetically engineered plant, we would regard as a food ingredient. This is sort of the legal construct, whether, if you're adding genetic material and it's expressing a protein, or you are changing deliberately changing the composition of the crop, that's sort of the piece that we treat as the ingredient under our ingredient authority. And we would evaluate it using the same kinds of information that we would use to evaluate the safety of any ingredient. This isn't limited to new proteins, as I mentioned earlier, although certainly those are a prominent feature of this category. So this is really our first question. The one caveat I want to make here, which was also mentioned earlier, is that if the protein is used to confer resistance to a pest, that's EPA's purview, and we would not look at that. We would focus primarily on the second question. The second question, of course, what else might you have done? So this is the sort of unintentional changes piece. We have authority over whole foods and the authority to make sure that they are not adulterated, and so that's what we're interested in here. And so the question we're asking is about differences, but of course, as I'll talk about a little bit later, it's whether those differences are relevant to safety that really ultimately matters. So back to that lovely flow chart again. You can use the 92 policy and the two questions that it identifies and all these peripheral flowcharts that are also in there to design a strategy for safety assessment. And in that strategy, you're essentially going to need to put together evidence to show that the things that you've deliberately introduced into the plant are safe and that the plant itself is as safe as comparable varieties. And so those are sort of the two piles of evidence that you're going to generate. In terms of figuring out whether what you've added is safe, there's obviously a lot of different information that goes into that. Again, this is really case by case. It depends on what you've introduced, what information is going to be relevant. In some cases, in many cases, all of these disciplines will be relevant. Sometimes other information can be useful. It really depends. If you've introduced a new metabolite, then that's obviously going to be important to consider that. And particularly when you're considering something you've introduced into crop, it's very helpful to be able to relate that to something with an existing history of use. That's whether it's similar or it's the same. That's very useful. A trivial example is something like oleic acid. If you introduce that, there's an extensive history of safe use. You don't really need to be too concerned in making your case that that's safe. But you can, of course, introduce more exotic metabolites or things that are not identical to things with an existing history of consumption. In those cases, there's a higher burden for experimental evidence to show that the presence of the substance in the transgenic plant is safe. I think it's also worth mentioning, again, in light of Wayne's talk earlier, that if you've introduced something that has sort of multiple effects or broader effects, something that could have pleiotropic effects or, as he pointed out, bring together two different metabolic pathways that weren't currently together, then you need to think a little bit about what the potential consequences of that might be. Again, not as a fishing expedition, but just to consider what are predictable or potential consequences of the substances that you've introduced into the plant. Since this meeting is co-sponsored by the Protein Allergenicity Technical Committee, I thought I'd talk a little bit more about safety assessment for proteins. So there's, as you'll see if you look at the 92 policy, there's really only a few things to think about when you're considering the safety of introduced proteins. The first, most obvious one is just the function. What is it actually doing? There may be some cases in which that's relevant to a safety or nutritional issues. But of course, in many cases, we have experience with that particular class of proteins, and it's not likely to pose any serious safety concern. One fairly generic example is just considering many enzymes that we've had experience with, and they typically have not posed a safety concern. So function. Toxicity. This is something that came up earlier. I think most people in the room know Most proteins are not toxins. There are relatively few of them. They come from a limited number of families. They tend to be acute. This simplifies safety assessment somewhat. If you have an identified significant similarity with a known protein toxin, you're probably in the clear there. And the final one is allergenicity. And here, one of the most important factors is just the source and the history of exposure. If you don't have that, or if you want to complement that, it can also be very valuable to analyze similarity to known allergens and protein stability, points like that. We do think that the approach that we tend to use is scientifically consistent with the consensus codex document on allergenicity assessment. In terms of that second question, what you're really trying to figure out here is, obviously, most plants, including agricultural plants, have low levels of toxins or anti-nutrients. And so what you're really trying to establish is just, is this as safe as the existing variety and not some sort of standard of absolute safety? So comparative analysis of toxicants and anti-nutrients is helpful. In addition, just because there may be expectations about the nutritional content of a particular plant variety, it's helpful to do a broader compositional analysis to try to get a sense of whether those expectations will be met for this variety. But we are aware, as has been alluded to several times today, that there's a lot of variation in terms of composition in plants, and so this background of variation also needs to be taken into consideration. What we're really trying to find out here is just the bottom line of differences that are relevant to safety. So starting point is trying to identify potential differences, but what you're really ultimately interested in, is this relevant to safety or nutrition in a way that would influence the outcome? So it's really a case-by-case assessment, and you're trying to establish whether or not the new variety is as safe as the old. The outcome of all this work is to put together a science-based narrative, and the narrative essentially explains why there are no safety or regulatory issues that would require further review by FDA, and it's going to be supported by relevant scientific information on the safety of your transformation event. And then once you've got that narrative, you would come and talk to FDA. As I mentioned earlier, we really encourage people to come and talk early and often. The initial process can be fairly free-form, and once the discussion has evolved to the point where the developer is satisfied that they have a compelling scientific narrative about why there's no safety or regulatory issues, they can initiate a final consultation process. And when FDA has no more questions about that submission, then the process is complete. Early and often is so useful because we can give feedback early on before people have committed to doing particular kinds of experiments. Because we do this case by case, we don't have a lot of detailed guidelines about what to do when. It really depends on the properties of the trait and the crop. It's especially valuable when the trait or the protein is novel. We even have a formal process called early food safety assessment or new protein consultation where companies can come in with a relatively novel protein that they're considering using in commercial transgenic crops and actually get feedback from us in the form of a letter about whether or not there are likely to be issues with that. There is no bar to companies coming in as early as they want, even at the product concept stage. It's free. We are happy to talk to people as often as they like. We can do it via teleconference or direct video conference or in person. So we really see a lot of value in developers discussing these issues with us as often as they think useful, again, rather than sort of doing a whole bunch of work and then bringing it in and then sort of this mutual discovery that's not actually what might be needed to complete the consultation process. The final consultation, as I said, it begins with a scientifically supported narrative about why there are no regulatory or safety issues that would require evaluation by FDA. It typically is based on specific transformation events and contains evidence about safety of the introduced protein or trait, evidence that the variety is as safe as comparable varieties, and identifies any material differences. And then the consultation is complete when FDA's team of review scientists is satisfied with the state of the submission and has no further questions for the developer. Once the consultation is complete, we post it on our website. We post both the letter and the note to file, which is a scientific memo describing our evaluation of the submission. And this is a really useful resource, both for the general public as well as for developers who may be new to FDA, to try to understand the kinds of things that we've looked at in the past and the kind of information that we've seen. So just to reiterate, you know, I think everybody sort of has mental checklists about the kind of information that is useful, but the bottom line is it's really whatever is needed to answer those two key questions we have about the safety of what's been added and whether or not anything has changed in a way that's relevant to safety or nutrition. So I just want to leave you with two things. The first is early and often. Please remember that and come in and talk to us. And the second is just to remember those two key questions. Our authority over food ingredients and over whole foods are sort of the underlying support for those questions about what you've added and what else might have changed when that happened. And finally, I just want to point out that we have two easy to remember URLs. This first one, GE Plant Foods, is for our main biotechnology page. And the second one, the Biocon inventory, is for our inventory of consultations. That's it. Thank you very much.